So sickle cell anemia, this will be our last, I think, presentation here on genetics. Sickle cell anemia results in irregular blood cell shape. The normal hemoglobin is designated as HB super A. Sickle cell hemoglobin is designated as HB super S. If you have the HB super A in the homozygote form, you're going to have normal red blood cells. If you have the HB super S in the homozygote form, you're going to have sickle cell anemia. And if you are heterozygote, have one of each type, you will have both normal and sickle cell or red blood cells, RBCs, red blood cells produced. Okay, so this is a picture here of the sickle cell shaped and the normal red blood cell shape. As you can see, the little sickle cells have corners, edges on them. They're kind of half moon shaped, and those points can catch on things, catch on if you've got arteries that are starting to clog, veins are getting plaque deposits in them, then you can have that catch as the blood flows past. It can catch on some of that stuff. They also catch each other. So when a person has sickle cell anemia, they've got two problems against, well, two or three problems against them. Two particular ones. First of all, the sickle cell blood cell doesn't carry as much oxygen as the normal red blood cell. So you have a problem getting enough oxygen to the tissues of your body, the organs. And then also, as you age, you have a greater chance of having clogging up of the blood in a vein or artery. Okay, somewhere it could be, it could lead to either a heart attack or stroke. So depending on where your blockage happens. So you've got a problem with that. Now, I am not an expert on sickle cell anemia at all. Okay, done some reading. So what I've read, and what I've been told too by some students who've had family members with some of this too. This is a inheritance trait that if you've got it in the homozygous form, the HB super S, HB super S, this is one where historically you would not live to reproductive maturity because you will die of organ failure due to lack of oxygen getting to the tissues before you're old enough to reproduce and pass on the trait, generally speaking. But today, in modern medicine, we have the ability to help people overcome for a little bit. Now, a number of years back, I had a student who told me his sister had that in the homozygous form. She had to take shots, and it wasn't hemophilia. That's a shot thing too. And I clarified that with him, and it wasn't. But I think she had to have some other types of treatments, and I don't remember the details, and I'm not sure. I didn't pry, and he didn't give me details. But they were, the doctors and the family were just very happy that she had made it to her 25th birthday. At that time, just recently had her 25th birthday with this, because that that was just not expected. Okay. So historically, that would have been removed from the population early on, which decreases the amount of HB super S that's going to get passed on each generation. The way that the HB super S survives in the population is in the heterozygote. And that's the HbA, HbS there at the bottom. So you have both types of red blood cells being produced. You think by, sometimes it's being referred to as a co-dominant, you'd have both at the same time, and maybe you do to some extent. But my understanding from what I've read of this is that the body actually will produce more of the normal unless you're under stress. When the body gets stressed, it switches to producing more of the sickle cell, if not exclusively sickle cell. And the reason historically for this is that this is a gene mutation that was naturally selected for in areas where malaria is prevalent. Okay, this gives a natural resistance, ability to survive malaria. So we've talked about malaria before back in our Kingdom Protista. I had you learn about the little Plasmodium vivax, our nice little single cell parasite there. It gets into the blood cell and reproduces, does its reproduction thing, and then it causes red blood cell to burst, releasing toxins out into our bloodstream, which result in fever. And small amounts of that we can survive; it's okay. But large amounts of that sends us into shock, and that's what eventually kills the person. For some reason, and I've speculated what that is, but I don't think I've ever read anything that explains it to me. Uh, that uh, what we do know is the little parasite gets into normal red blood cells, but not into the sickle cell shaped ones. And I don't know if that's because there's something about the cell membrane that is thicker. Shape is different, so it could be that the membrane has a slightly different structure. Um, those phospholipids may um, I don't know, be tighter together, more selective about what gets in. I don't know. I'm not sure what's going on there, but it doesn't seem to get in there. Or if it does get in there, maybe we just don't realize it gets in there. It can't live. And well, why wouldn't it be able to live? Is it just not enough room or what? Well, if these little cells can't carry enough oxygen to keep our tissues alive, okay, over a long period of time, maybe they don't carry enough oxygen for the little Plasmodium vivax to live, okay, and survive and reproduce. So maybe that's a factor. I don't know. Those are my speculations. It's got to be something. Okay. Anyway, someday I'll find out. I think I'll run across the article that'll tell me. But anyway, historically, what I have read is that historically, and still today, but we'll go to historical context on this. When someone got malaria, that causes stress on the body. The first few blood cells that burst and those toxins go out there and cause our body to react with fever and things like that. That's stress on the immune system. And so the body would start producing more sickle cell shaped blood cells. And what that does is that reduces and or even eliminates the number of normal red blood cells in the bloodstream. And so the resulting little parasites that have, the reproduction, been reproduced and are now in the bloodstream can't find a home. Okay, there's no red blood cells for them to go into other than these sickle cell shaped ones, and those aren't good. Doesn't work. When the stress went away, basically when all little parasites died and couldn't do anything more because they couldn't find a cell to live in, then the body would go back to producing normal red blood cells. So the problem with this today is that people with the heterozygote, the HBA, HBS genotype, they don't seem to, well, I mean, look at society today. So when does the stress stop, right? As a student, when did your stress stop? Anytime during the semester? Okay, it just gets worse. And school's over and you're like, breathe a sigh of relief and it's something else, right? You've got other things in life causing you stress. If you work, your job's a stress, your career's a stress, everything's a stress. You get older and you get kids, so that's a stress. And so stress never lets up. So once your body kicks into these producing the sickle cell blood cells because of stress, there's the trigger for it to go back to producing normal doesn't seem to be there. Okay, according to what I've read, that's what happens, is that this really gets tough on people at about age 45 into the 50s is where it triggers really bad, or triggers before that, it shows up at about that time. They're under a lot of career stress. They're under a lot of child stress. If you had kids younger, that's about the time they hit teenager or go off to college. If you had children later, well, you've still got young kids at home, and the stress doesn't go away. And so we start seeing at that point, after a few years, it builds up, and people in their mid to late 40s and early to mid 50s have heart attacks. They have organ failure, tissue problems starting to cause organ problems. Okay, historically, 500 years ago and before, and even maybe more recent than that, it wouldn't have been a problem because people got married at 16, 17, 18, had kids. By the time you're 45, you're a grandparent. which we like to keep our elders around, but biologically speaking, as far as nature goes and survival of species, you've done your job. You reproduced, you got your kids up old enough they can reproduce. And so if you die of something like this, hey, you survived malaria, so you could reproduce. And that's all that's important for survival of the species. Okay. Not for maybe well-being of your family, but for survival of the species. Okay. So that's how natural selection played a role in this, but how it also has a downside as far as longevity and more modern day society where we live older, we tend to live older, we have expectations of living older, we have expectations of having grandparents and great grandparents around. Okay. All right. And people in that age group are not old and retired and on death's door like they might have been 500 years ago. Okay, people in their 40s and 50s are still working, productive members of society, and in sometimes, most times, they're at the peak of their career. They're contributing the most to their jobs and their careers that they have, and probably and possibly will in their career. Okay, so looking for treatments and cures and things for that is a part of genetics, and looking at some things where we could go in. What causes this? If you remember back in my DNA talk, back in the chemistry of biology, when we were talking about DNA, I briefly mentioned if you go back and you look through those slides, you're going to find, I believe, I don't think I took it out, I think it's still in there, where this is just a single base mutation. Okay, in the middle of one of those codons, okay, well, it wouldn't be a codon in DNA, but for the RNA, we show that so there's the little, 3 bases that code for a protein. In the middle of one of those 3 bases, one of those triplets, there is a mutation of a single nucleotide, and it goes from, I don't know, I should have looked this up, but it's like a G is swapped for an A or something like that. Okay, and it changes the amino acid that is put into the protein. It's kind of in the middle of the whole thing. And in some cases, that would make when that happens, as we've talked about before, that would make that protein totally inactive. It wouldn't be able to do what it's supposed to do. If it were an enzyme, it wouldn't. It would trigger whatever reaction it's supposed to start, or it wouldn't stop whatever reaction it's supposed to start. It would not turn on or off its process or do its thing, be the building component that it is. Okay. And here it doesn't stop the protein from functioning. It does go ahead and allow it to be a component of the red blood cell and function somewhat, but it is that one nucleotide change causes this entire change of shape of this red blood cell. Okay, so that's kind of the impact that just one nucleotide change can have, other than being detrimental. Okay, this one is kind of detrimental. Okay, it's not fatal. It doesn't keep the organism from living, but under certain circumstances, it's detrimental. And another perspective, it's advantageous. Okay, if you live in an area of malaria, this is a natural resistance way to survive malaria. Okay, epistasis. This is where a particular gene combination hides the effect of what would normally be dominant. So this is, we're going to use mice. Epistasis is mice as an example. So we're looking here at mice with either brown or black being the dominant, coat color. Depending on where you're reading and what book you're looking at too, this can sometimes be called brown and tan or black and gray. Okay, there's some different distinguishing. Field gray, I think, is one of the field mouse is a field gray. But anyway, we're going to go with black and brown because that's what our illustration here has, and it's a Punnett square. It's turned on its edge. So it looks like I would call it a Punnett diamond if you want. I don't know. You just kind of turn it in your mind. But this is your dihybrid. We're following 2 traits. We're following the 2 alleles for color, capital B for black and little b for brown, and we're following a secondary set of alleles that's going to determine whether that color gets to be expressed or not. So if you inherit, and the C's, if you inherit a capital C, okay, an allele with a designation of capital C as a dominant there, then that allows the pigmentation in the previous pair to be expressed. Okay, so that brown or black color that you inherited will actually get shown. However, if you inherit the C allele pair in the recessive little c state, then it doesn't matter whether you've inherited black or brown from your parents, you're not going to express any pigmentation and you're going to be albino. That's our little white laboratory mice. Okay, those are albinos. And that C can affect pigments other than hair color. Okay, same type of thing happens with eye color. Okay. In humans, you get skin color, you get hair color, you get eye color all affected, and no pigmentation shows. Red eye is actually not red eyes. The iris of the eye is colorless, no pigment, it's clear. So what you see is the light comes in, reflects off the back of the retina, the blood vessels back there, and what you see is the pink look, pink or red look is from reflection off of blood, light off of blood. Okay, and because there's no iris there to filter that light back through, so you see a color. Okay, it's just there's no filter there. It's all clear, colorless. Okay, so that's an example of epistasis, and that's albinoism is an example of that. Last bit here is somewhat of a review from plants, and I know pretty sure also before we left on spring break, back there somewhere I pulled out the flower model and was talking to you about pollination. I was talking to you, I know we weren't talking about plants yet, but we're talking about other things. And we were talking about double fertilization. Somehow I believe that was mentioned. If not, that is, I do talk about it. I don't really show you as much, but I talk about it in the plant presentation, so you can go back and watch that. But to review some terms and to make some distinctions here. Pollination and fertilization are not the same thing. Pollination is where you get the pollen onto the female part of the plant. Okay, transfer of male pollen grains to the pistil of the flower. Pollination can happen between two plants that aren't even the same species. You can get pollen from anything onto that pistil of the flower, but that doesn't mean anything. Okay. Now, if pollen of the right species gets on there and grows a pollen grain down into the ovary of the flower, of the pistil, then you can have fertilization occur. Okay, fertilization is going to be where that pollen grain releases sperm down through the pollen tube into the ovule, and okay, the pollen grain or the sperm has to unite with the ovule, the egg. Egg and sperm have to unite. That's fertilization. Okay, all that has to happen before you have fertilization. Okay, so pollination does not guarantee fertilization, nor does it guarantee seed production. All right, that ovule, that fertilized ovule will mature into a seed later. Okay, and we talked about all that in plants. So self-pollination, of course, is where the male and female gametes come from the same plant. That means the pistil can receive pollen from the anther in that same flower. Okay. And I think I was kind of talking about how the anthers in the model in my lab, my classroom, the anthers were above the top of the pistil. So that's really easy for it to just kind of fall down in there. Insects can brush it in as they walk past or fly past going down into the flower for nectar, this and that. Wind can just dust it there. Some plants have the anthers lower down below the top of the pistil, and that's going to cause a little bit of difficulty in self-pollination, though it can still happen. Okay, it's going to rely though upon excuse me, usually an insect or something to carry it, transport it out. Usually those flowers with that structure are designed to not self-pollinate. They are designed to cross-pollinate. Another thing though, cross-pollination can occur even if the anthers are in a position where the pollen could fall onto the pistil. Just by the anthers producing their sperm or their pollen at a different time than when the ovule is ready to be fertilized, either before or after. And what that means is that that pollen is going to have to be picked up on the hairy legs of a bee or a fly or the hairy wings of a butterfly or something and carried off to another flower that is either flowered a day or two earlier, okay, and now the ovule is ready to receive the pollen, the sperm, or be carried to a flower that is just now freshly bloomed out, and the ovule's ready to receive, and the pollen's not mature yet. I mean, there's just the different maturity times of the two will guarantee that cross pollination has to occur in those particular plants. And of course, I talked about things being transferred. Those are called the things that transfer the pollen from one plant to another, referred to as vectors. It's a term you'll probably see it on the test. And that can be insects, as I mentioned, wind, humans, and other animals as we walk through grasslands and places we can brush past. And if you've ever gone walking out a field in late summer in particular, and probably drive your hay fever nuts if you have that, you'll pick up all sorts of yellow green dust all over your pant legs. Um, that is pollen. Okay, and you've picked it up off one plant. And you say, well, I didn't dust any off on another plant because I took it all out of the field with me. But you did. You picked it up off one plant, dusted off on another plant, and picked up some more. Okay, and that's just what the animals with hair, fuzz on their legs and wings do. Okay, they pick up some, next plant they brush off some and pick up some more. So we're all vectors, we travel carried around. And of course that's male parts there, female parts there. We've had that already in plants, so I won't go back over it. Okay, that should do it for genetics. There may be, I may go ahead and do a, I tried probably do an abbreviated version of mechanisms of evolution. If I do it all, that may get dropped off due to time constraints here at the end. So just look on your D2L and see if that's visible or not, or if I've taken it off.